आप लोग बात मत करिएगा बेटा, है ना। नमस्कार, आप सबका आंचलिक विज्ञान केंद्र भोपाल के इस प्रेक्षागृह में बहुत बहुत स्वागत है। एक बार इस प्रदर्शनी के लिए, खास इस अभियान के लिए जिसे हम साइंस फिएस्टा कहते हैं, अभी आप इस व्याख्यान के बाद ऊपर जाके, आज इतना बढ़िया व्याख्यान देने वाले हैं प्रोफेसर संजीव शुक्ला। आप उस व्याख्यान के बाद जब ऊपर जाएंगे प्रदर्शनी में देखेंगे तो बड़े बड़े जो वैज्ञानिक और जो रिसर्च स्कॉलर्स आए हुए हैं, अपने अपने अनुसंधान या प्रोडक्ट्स को अपने साथ में लेके, आप उनसे इंटरेक्ट करिए, उनसे बातें करिए। आपके मन में जो भी प्रश्न आते हैं या उस इंस्टीट्यूट से जुड़ा हुआ क्या वो काम करता है, आगे अगर आपको उसमें कैरियर बनाना है या उस फील्ड से रिलेटेड जो भी सवाल हैं आपके, आप पूछिए बेहिचक। आप सब इन सब बातों को ध्यान से पूछेंगे कि नहीं, हाँ या ना? मुझे लाउड एंड क्लियर, ताकि हमारी यूट्यूब पे जो ऑडियंस अगर देख रही होगी उनके लिए भी आवाज पता चल जाए, हाँ या ना? एक बार और। थैंक यू सो मच। तो वी हैव विद अस टुडे प्रोफेसर संजीव शुक्ला जी। ही इज दी वन ऑफ द ग्रेटेस्ट साइंटिफिक इंस्टीट्यूट्स के एक फैकल्टी हैं, और वो उनकी वाइडर रिसर्च है, वो कैंसर पे भी काम कर रहे हैं, और आज आपसे जीनोम्स के ऊपर बात करने वाले हैं, और उससे जुड़ा हुआ कैंसर्स के बारे में भी सर अगर उस पर रूबरू आपको कराएंगे। मुझे खुशी है कि सर ने हमारे आमंत्रण को एक्सेप्ट किया और आज आप सबसे बात करने के लिए इंडियन इंस्टीट्यूट ऑफ साइंस एजुकेशन एंड रिसर्च भोपाल से समय निकाल के हमारे बीच में हैं। तो एक बार जोरदार तालियों के साथ आज के हमारे स्पीकर सर का हम स्वागत करते हैं। और बहुत जल्दी मैं सर के बारे में बताता हूं। सर का पीएचडी एडवांस सेंटर फॉर ट्रीटमेंट रिसर्च एंड एजुकेशन इन कैंसर, एप्ट्रक जिसे बोलते हैं, ये जो कि टाटा मेमोरियल हॉस्पिटल है बॉम्बे में, उसका एक बहुत बड़ा R&D रिसर्च इंस्टीट्यूशन है मुंबई में। सर ने वहां से अपनी पढ़ाई की है, और उसके बाद सर वेलकम ट्रस्ट और डीबीटी यानी डिपार्टमेंट ऑफ बायोटेक्नोलॉजी के फेलोशिप के साथ उनका कंटिन्यू हुआ है, रिसर्च वर्क प्रोजेक्ट्स में इन्वॉल्व हैं, और सर का काफी लंबा कैरियर है। सर के कई रिसर्च पेपर्स हैं, और सबसे बड़ी कमाल की बात, इन सब के बावजूद भी सर टाइम निकालते हैं और स्टूडेंट्स और बच्चों से इंटरेक्ट करते हैं, ताकि वो फंडामेंटल साइंस में जो कंट्रीब्यूशन है, जो काम हो रहा है, वो आप तक ले जा सकें। समय के अभाव में हम ज्यादा समय नहीं लेंगे, आपसे यही कहेंगे कि इस प्रोग्राम के तुरंत बाद आप जाके ऊपर हमारे साइंस फेस्टा का आनंद लें, एग्जीबिशन का, आप एग्जिबिट को इंटरेक्ट करें, जो भी साइंटिस्ट साथ में आए हैं अपने अपने इंस्टीट्यूट से। और अब मैं सर को आमंत्रित करने के पहले अपनी टीम के मेरे कलीग से ये जी हाँ, आप ही करिए, आइए ठीक है सर आइए। यहाँ सर, यहाँ प्लीज, लाइव भी जा रहा है प्रोग्राम। सर आप दीजिए जी। जी आप सब जिसके लिए इंतजार कर रहे हैं वो समय आ गया है। अब मैं सर से दरख्वास्त करूंगा कि सर, मैं आपको इंट्रोडक्शन बहुत ब्रीफ दिया हूं समय के कारण, लेकिन आपका ये मंच है और आप बच्चों से इंटरेक्ट करिए। आप देखिए इस ऑडिटोरियम की कैपेसिटी से आई थिंक 2.5 टाइम्स से भी ज्यादा लोग हैं, सब जगह पूरा ऑडिटोरियम भरा हुआ है, इवन डाइस पे बच्चे हैं। तो स्टेज इज योर्स सर।
So, good morning to all of you. I'm really happy to see a room with full of energy, full of young energy. And so basically what I'm going to talk to all of you today about is beyond the genome, from development to diseases. You must have studied in your school about the genetics, about the genes that make the protein, which then perform all the functions in the cell. I'm going to tell you today about beyond the gene, what are the factors which matter, and genes are not all your destiny. You can actually, it's also bit philosophical talk as well, because I will tell you that you are not what you just inherited from your parents in terms of DNA. You are more than, much more than that, right? So if you look at this slide, you will see that we inherit a lot of characteristics, lot of features from our parents, such as the eye color, the ears, the size of the ears, the height, various features that are inherited from the parents in the form of DNA, right? So it's the ATGC which basically determine who we are going to be, right? So there are almost 3,000 million letters that we receive from our father, and the same number of letters we receive from our mother, right? And that's what makes us who we are today. And that's why when the first draft of Human Genome Project was announced, there was a lot of enthusiasm, lot of excitement about that now we understand everything, now we can now we know how the biology works, now we know how each cell of our body works, and we can cure all the diseases because now we know what is the DNA sequence, right? So there was a lot of over enthusiasm. In fact, at that time, then the President of America, he said, we are learning to the language in which God created life. But slowly we learned that actually learning the DNA sequence is not enough, because there are a lot of things which are not inherited by the DNA. You do not inherit how whether you are going to have good manners or bad manners. You do not inherit which language are you going to speak. It depends on your surrounding, right? You can speak Hindi, Tamil, Telugu, Kannada, any language, it doesn't matter. You learn from your surrounding. It's not in the DNA. Whether you're kind to others, whether you have a good sense of humor or bad sense of humor, all that is not determined by your DNA, right? It depends on your environment. So can you hear me? All right. So so basically what I'm trying to tell you is that there are things beyond the DNA which make us who we are. And if we look at the journey of the life, it basically starts from a single cell, single fertilized egg called zygote, right? So after the fertilization, the sperm and egg join together, make the zygote, make the fertilized egg, and this zygote then further divides, makes the embryo, and then the baby is born, right? Now, if you look at the DNA of the zygote versus any of the embryo cells or any of the cells from this young born or any of the cells from the adult, DNA is exactly same. There is no change in the DNA sequence, right? And it's basically the 6 feet long DNA which is packaged in our cell in the tiny 10 micrometer nucleus, right? So you can imagine that a 6 feet long DNA has to be packaged inside a very small territory, right? So it's like it cannot be packaged like a wool ball, right? Otherwise, what will happen is if you need any part of the DNA to be expressed in a particular cell type, it will be very difficult to bring it out, right? So there is a very nice packaging of the DNA in every cell type that determines what are the genes that are going to be expressed in one cell type versus another cell type. So if you look at any of the cell type, DNA is exactly same, but each cell type knows what it's going to be. For example, if you look at the different adult stem cells, they know what's their fate going to be, right? For example, myoblasts cells, they know that they have to form only muscles. Keratinocyte cells, they only form skin cells. Hematopoietic cells, they know they will only form blood cells. So they do not forget what they are going to be, right? Although if we look at the DNA from myoblasts, keratinocyte, hematopoietic cells, all have exactly identical DNA. So how does it work? Does anyone know how does this work? How do our different cell types like skin cells, eye cells, liver cells, how do these look different? How do they perform different functions? Anyone has any idea? Anyway, let's continue. So this is what we are going to learn today. So beyond the genetics, beyond the genome, there is epigenetics, and that is what basically determine various things that I am telling you today. So epigenetics is basically above or in addition to genetics. So modern definition is non-sequence dependent inheritance. So it does not depend on the sequence of the DNA. It is above the sequence of the DNA. And by learning epigenetics, we can answer several questions. For example, how can identical twin litter mates show different coat colors? Or if we take it from the human perspective, even the identical twins, they do not behave identical, they are not susceptible to the same diseases as they grow up. They are born exactly same, but as they grow up, they become different individuals. They acquire different chemical tags, they acquire different epigenetic marks, which makes them different. How can just paternal or maternal traits be expressed in offspring? So there are traits, there are genes, so each gene, basically there are two alleles in our chromosomes, right? All of you know that? Do you know that whether both the alleles are expressed in all the cells? Say for example, can you name any gene? Right? So both the genes, in most of the cases, both the alleles are expressed. That means both the chromosomes are expressing that same gene. But there are genes which are not expressed by both maternal and paternal chromosomes. That means some of the genes are expressed only by the chromosome that are inherited from mother, and some of the genes are expressed only from the chromosomes that are inherited from father, right? So if you do not have this combination, there will be embryonic lethality. We cannot survive. That's what the complementation is. Cannot survive. That's what the complementation is. As I will talk to you in coming few slides, how does it impact? The next thing is, how can females express only one X chromosome per cells? We all know that females have two X chromosomes, males have only one X chromosome, right? Does it mean that females will have two copies of their genes expressed from the 2X chromosome, and that means they will have more amount of the X chromosome related proteins, right? But that doesn't happen. Nature has balanced it out by allowing only one chromosome to be expressed and the other X chromosome remains silenced. So there is equality in terms of the expression of X chromosome genes in males and females. Males have only one X chromosome, females have two, but only one of the X chromosome of female will be expressed. Who determines that? It's the epigenetic changes which determine which X chromosome is going to be expressed. How can acquired traits be passed on to the offsprings? Are there traits which can be passed on to the offsprings beyond the DNA? We know that we inherit the traits from our mother and father in the form of DNA. But are there traits which can be inherited beyond the DNA? That's what epigenetics tells you. So basically epigenetics is a term which was coined long back in 1942. And at that point of time, basically, there were two branches, development and genetics. Genetics was to understand how the genes function, and the development was to understand how the single cell, how the after the fertilization, how the zygote gives rise to the whole individual, how the developmental stages are regulated. But whatever could not be understand at that time, based on the genetics or based on our understanding of development, a new term was coined that it is happening because of epigenetics. Although at that time it was not clear how does it work, there was not a good understanding that what are the mechanisms that determine these epigenetic changes, but a term was coined and epigenetic landscape theory was given, which says that at the time of the fertilization, all our cells are at the top of this hill, right? So basically, just consider it as a zygote. Now, as the zygote divides, makes the embryo, there are a lot of the embryo will then differentiate into different lineages. There will be some cells which will make muscles, there will be some cells which will make liver cells, brain cells, and all those different kinds of cells, right? So as the cells differentiate, this ball rolls down and it goes into different troughs, right? And as the cells continuously differentiate and comes down to the bottom of this hill, it becomes completely differentiated. So it starts from undifferentiated stem cell. By the time it comes down the hill, it becomes completely differentiated. And that's why going down the hill is easier, going up the hill is always difficult. So that's why these differentiated cells, they do not de-differentiate in the normal condition. So this epigenetic landscape, although this was given long back, but this still holds true even in the current date. So basically, our DNA is not naked. It is wrapped around several proteins, which are called histone proteins. And beyond the DNA sequence, what is that is determining the different phenotype is basically the chemical tags which are present on the DNA as well as the proteins. So the chemical tags on the DNA are called DNA methylation, also the various derivatives of DNA methylation, and various chemical modifications that can take place on these histone proteins, which are called histone modifications. So altogether, these modifications determine whether our DNA is going to be packaged tightly or it's going to be packaged loosely. So what happens is, if it is packaged tightly, what will happen is our transcription machinery will not be able to access that part of the DNA, and that part of the DNA will remain silenced. That means those genes will not be expressed. For example, if you take myoglobin, myoglobin is expressed in what kind of cells? Which cells express myoglobin? Muscle cells, right? Very good. So muscle cells, the myoglobin gene will be in relaxed condition so that the transcription can happen, it can express. But if you go to, say, liver cells or neuron cells, myoglobin gene is still there, but it does not express in those cells. So what happens to the gene? It is tightly in a compact form so that the transcription machinery cannot access this myoglobin DNA, and it does not express the myoglobin gene, right? So whichever gene needs to be expressed, the DNA is in the relaxed form. Whichever gene needs to be suppressed, that DNA is in the compact form. So all of you know this central dogma that DNA gets replicated during the cell division, and during the transcription DNA forms RNA, and this RNA is translated to give rise to protein. Have you heard about this central dogma of life? Okay, so life would have been very simple if it was just plain DNA to RNA to protein, but as I told you in the last slide, that our DNA is wrapped around proteins, and there are modifications which decide whether the DNA is going to be compact or relaxed. So, as I told you in the last slide, if the DNA is compact, the gene will be silenced. If the DNA is more relaxed, the gene will be expressed. But life is not so simple. If you look at this slide here, what you are seeing is there is one inactive gene where the DNA is compact, and there is a highly expressed gene where DNA is more relaxed. But there are several, like there are several shades of gray, the gene expression can also be fine-tuned. It's not that it's just going to be on or off. It can be expressed as per the need of the cell. If you need more protein, it is more relaxed, more amount of mRNA will be produced. If you need less protein, it will be little bit relaxed, and less mRNA will be produced, less protein will be produced. So, a fine tuning of the gene expression can be performed by epigenetic regulation of gene expression. So, I'm sure all of you like movies, right? So, if we consider, if we just have an analogy between our human life and movies, the actor and actresses of our life are basically the cells. The muscle cells, liver cells, skin cells, brain cells, all these are actors and actresses, and the DNA is the script. So script has all the instructions for all the participants of the movie, what role are they going to perform, everything is written in the script. And same script is given to all the actors and actresses of that movie, right? So that means same DNA is given to all the cells of our body. There is no change in the script. And if you look at the gene, it's kind of the dialogue in the script, and bases are basically ATGC, they are the words in the script. Now, who decides that which dialogue is going to be spoken by which actor? If it's like same script is with everyone and everybody speaks the same dialogue, it will be a disaster, right? So the concept of genetics is like screenwriting. So genetics is screenwriting, everybody has the same script, but is the director who decides which actor will perform on which dialogue. So likewise is the epigenetics, which acts as a director of this movie, our human life, and decides what sets of the genes, what sets of the dialogues are going to be spoken by which cell. So it's like a radio jockey or DJ, which sets of the songs are going to be performed in which event. Likewise, which sets of the genes are going to be expressed in which cell type is decided by the epigenetics, right? So there are two mice you see on the screen over here. What is the difference that you see over here? Color of the fur is different, right? Size is different. One is obese, one is normal in size, right? And if I tell you genetically, both the mice are exactly same, 100% identical. There is not even a single base change in these two mice. So why do they look different? It's because the mother of these two mice was fed differently. Mother of this mice was fed with the standard diet along with all the vitamins and minerals, and mother of this mice was fed with only the standard diet but not with the vitamins and minerals. So what happened is, because of that, or just have a look at this. Can you click on this small click? Yeah. If you take the cursor to the center of this, it will come. So this is this obese mice, and this is the lean mice, right? Genetically, both are exactly same. But what is the difference? Because of the diet, what happens is, in this case, the mice was fed with all the vitamins and minerals, and these vitamins and minerals, they are used as a cofactor for these epigenetic enzymes. DNA methylation is one such epigenetic modification, right? So what happens is, there is a gene called agouti gene, and because it had the cofactors which are required for DNA methylation to happen, there is a DNA methylation that happens at the promoter of this agouti gene. And because of this, agouti gene is silenced. So DNA methylation is associated with silencing of the gene in general. But in this case, because the mother of this mice was not fed with the vitamins and minerals, the methylation could not happen at this gene. So this gene was highly expressed, and because of this high expression of this gene, it correlated with the obesity, and this mice was susceptible to various diseases, including cancer, cardiovascular diseases, diabetes, and so on. So you can imagine that the diet can make such a huge difference in determining what's the phenotype going to be, right? So, and it's not that it is irreversible. So if you, again in the next generation, if you feed the mother with standard diet and along with all the vitamins and minerals, again you will have a normal pup, normal mouse, right? So that is, these are the maternal supplements, the zinc, methionine, choline, folate, B12, and you can relate it to the humans as well, that during the pregnancy, mothers are given additional supplements of vitamins and minerals to make sure that all the nutrients are given to the mother. Now, this example is again it emphasizes the role of epigenetics. So there is Burker V and the Queen V. Again, genetically, at the level of DNA, both these V's are exactly identical, but the difference is, again, epigenetic. And how this epigenetic differences come up is because of the royal jelly. Who gets the royal jelly? The Queen V gets the royal jelly, and royal jelly has lot of nutrients, and within these nutrients is there's also inhibitors for DNA methylation. Again, just don't worry about methylation, inhibitors for epigenetic changes. So what happens is, in the Queen V, the queen genes remain expressed because it is inhibiting the factors which are going to silence the queen genes, right? So because of that, the Queen V becomes Queen V because the queen genes are expressed. In case of Worker V, it is not fed by the royal jelly. So what happens is, these queen genes are turned off, and because of that, this becomes Worker V. So basically, in a way, you can say that you are what you eat. So you should make healthy choices. So I'll just show one quick clip here. Not coming. If you click on this. Oh, okay, it's up to this. Okay, anyway, another example of how diet can influence your gene expression is the Dutch hunger winter of 1944. What happened during this time is the Germans blocked the food and fuels shipment to the western part of the Holland from September 1944 to May 1945. So there was food scarcity at that point of time, and because of that, the children who were born at that point of time, they were more prone to developing diabetes, heart diseases, obesity, schizophrenia, and so on. It is understood that because the mother was not fed with the proper diet, the children might have suffered, right? But this did not limit to just the children, but also the grandchildren of the hunger winter mothers. So this again emphasizes that these epigenetic traits could be passed on from one generation to another generation in a similar way as the DNA sequence is passed on from one generation to another generation. So actually you can say that what you are today is not just the result of what is cooked in your mother's kitchen, but also your grandmother's kitchen, right? Can we have a clip now? Yeah, here. So you don't need to keep blaming anybody here, because the good thing is all these epigenetic changes are reversible. Genetics is not reversible, but epigenetics is reversible, so you can be what you want to be by not just adopting a good lifestyle, good diet, but overall good routine that you can have. So right now I just told you about the diet. Is the diet the only thing that affects the epigenetic changes? There are a lot of other things. It's also the care. It's also the love and affection that you get within your family. For example, in this experiment, if you look at this mother is very attentive, caring, and in the mice it's basically the mother licking the pups. It licks the head of the pups, and that's what this care mean for the mice, right? And what happens, so this is one group. Another group where mother is unattentive, does not care, they just ignores their child, right, their pups. What happens is, in this case, the hypothalamus there are genes which because of this leaking they are rarely methylated. So these genes are expressed in the puffs of the mother who is very attentive. But in this case, because the hypothalamus or the brain was not leaked, these genes remain highly methylated, and because of that these genes are suppressed. These genes are not expressed. And because of that, when this pup will become adult, these puffs will be more relaxed, and over here these puffs will be more stressed, more anxious. They will not be able to keep calm in any stressful situation. So you can imagine that not just the diet, but there are several things that can affect what you are going to be. And again, it's reversible. So you don't have to worry whatever, even if you did not get the care in the early childhood, that doesn't matter. By acquiring good lifestyle, good diet, you can still make changes. Another point, which again emphasizes the role of epigenetics, one experiment where the mice was allowed to smell the cherry, and immediately after the cherry smell, they were given an electric shock. So what happens is, after a couple of experiments, this mice became tuned to this, became habitual that whenever I will smell the cherry, I will be given electric shock. So it started feeling threatened whenever there was a cherry smell. Even if there was no electric shock given later on, just by the cherry smell, it became threatened. That's fine. But in the next generation, this mice was trained to get threatened, but the next generation mice who was never given any electric shock, by just bringing it close to the cherry, it became threatened. This experiment suggested that there could be actually features that could be inherited from one generation to another generation. There could be even the experiences that one acquire, they could be transmitted from one generation to another generation, right? Now if you look at the crocodile, by looking at these crocodile, can you tell me which one is male, which one is female? It's not possible, right? All right, even if I tell you that you take the DNA from this crocodile and now tell me which one is male, which one is female, can you tell me? If you take the DNA from your cells, can you tell, and don't reveal the identity of any of you, can we tell based on the DNA which DNA is coming from male student, which one is coming from a female student? We can tell because of the chromosomes, right? Sex chromosomes will be different. But in this case, the sex is not determined by the DNA. It is determined by the temperature at which the egg is hatched. If the egg is hatched at the lower temperature, the crocodile is going to be female. If the egg is hatched at the high temperature, crocodile will be male crocodile. Now, how does the temperature makes this difference? How does the temperature determine who is going to be male, who is going to be female? It's again epigenetic expression. So what happens is, at the lower temperature, there are a set of genes which are activated, which determine female crocodile, and at the higher temperature, those genes are suppressed, and there are other set of genes which are expressed, which determine that this crocodile is going to be male. So again, you can see that there is epigenetics in every spheres of life. It's not just in humans, in all forms of life. If you now look at this slide and look at all these different species, and if I ask the question, what's odd about mammals? One thing is common in all these species, but in mammals it doesn't exist. So they all can have parthenogenesis or virgin birth. What does it mean? It means that the male and female genome, they do not need the male and female genomes to come together to form the animals. They can have parthenogenesis, but mammals, they cannot have parthenogenesis. There cannot be virgin birth. There has to be sperm and there has to be egg which needs to be fertilized to form a mammal, right? Now what's the reason for that? Remember in the first few slides I told you that there are a set of genes which are expressed by the maternal chromosomes and there are a set of genes which are expressed only by the paternal chromosome. So you need both. So there are actually experiments, so it takes two to tango. There are actually experiments done where people have tried to fuse two sperm nuclei. In the case of these experiments were done with the mice, so they could not get any mouse. When they tried to fuse two egg nuclei, again they could not get any viable mouse. Only when they fused sperm and the egg nuclei, then only they could get the viable mouse. And the reason for that, see, you are getting the equal nuclear material from the two sperm nuclei and or from the two egg nuclei. So theoretically you should be able to get a viable mouse, but it doesn't happen because there is a phenomena called genomic imprinting, which what it does is it determines that there are going to be genes which are expressed only by the father's genome. There are genes which are going to be expressed only by the mother's genome. So you need both so that they can complement each other and you can get a viable mouse. So it's again that is determined by the epigenetics, not by the genetics, because DNA is there, but still it cannot express because of the epigenetic silencing of alleles from the mother's gene or the father's gene. And I will not go into the details of this, but just understand that there is a phenomena called genomic imprinting which determines that there are genes which are going to be expressed by mother or father. I was also telling you about one phenomena which is called doses compensation. Doses compensation is basically there are two doses of X chromosome in females and there is only one X chromosome in males, but still the expression of X chromosome genes remains same, and that is because immediately after the fertilization there is a phenomena called doses compensation which inactivates one X chromosome in the females, and that's how both males and females they express only one X chromosome. We talked about diet, we talked about the care and affection, we talked about basically various different things which affect epigenetic changes. But there is one more experiment that will tell you that even you might have heard from your school teachers also, your parents also, if you don't use your brain, basically use it or lose it. If you use more part of the brain, you become more intelligent, right? If you don't use it, it's basically going to remain unutilized. This is what is shown in this experiment. What they did is they allowed the mouse to remain in the standard condition or the impoverished condition. So basically just a cage, they were just locked in a cage where they gave the diet and everything, but there was nothing else to do. They were just locked in the cage. And these mice over here, they were also locked in a cage, but this cage was more enriched. More enriched means they gave various equipments to the mice so that mice is not just eating all day. It can also exercise, it can do, it's like a mouse gym was given to them. What happened is, at the end of this experiment, the scientist here, Marion Diamond, she looked at the cervical cortices of these, this experiment was done in rat, not mice. So she looked at the cervical cortices of these rats, and she found that the cortices were thicker in the mice which were in the enriched condition as compared to the impoverished condition. Which, what does it mean? Because these mice were more active, they were using their brain more, that's why their cervical cortices were thicker as compared to the routine cages. Also, you might have heard from the doctors that maybe you need to change your, you need to go someplace, say hill station, just change your environment and you will feel better. You know, people have done these experiments where they have looked at the gene expression at different locations, and they find that when people move from one location to another location, the same set of people, their gene expression changes. So again, emphasizing the environment, the surrounding affects the gene expression. All right, so now coming to this very interesting example, all of you know what is shown on the screen? This is the first clone, right? Dolly the sheep, right? And so basically this Dolly the sheep, where was it? Where was this experiment carried out? Roslin Institute, right? And who did these experiment? It's Ian Wilmette and Keith Campbell, right? What they did in this experiment? Why was this experiment so exciting for the whole scientific community? First clone ever made. What they did was very interesting. They took the egg from the donor sheep, right? And they enucleated. That means they removed the nuclei from this egg. And what they did is they took the memory cells. Memory cells are basically, I mean, they are already differentiated. They are the adult cells, right? Now these memory cells they took and they isolated the nuclei from this memory cell and introduced it into this enucleated egg. Now the surprising thing was that this enucleated egg, when this memory nuclei was given, the memory cell nuclei, it could change its gene expression and it could become it could form the embryo when it was transplanted into the surrogate Eve, it could generate another sheep. Why it is exciting? Because just imagine that the memory cell and the egg cell nuclei, as I told you, that at the level of DNA it is exactly same, but the genes which are expressed in the memory cells of this versus the genes which are expressed in the zygote, they are completely different. The fertilized egg will express different set of genes, memory cells is going to be express different set of genes. But because of the environment of the egg, the gene expression altered in these memory nuclei, and because of that there was basically the de-differentiation. Remember the epigenetic landscape I told you? The ball at the top of the hill versus ball at the bottom of the hill. I told you that going back to the uphill is always difficult. So it was actually going uphill task for the memory cell, the memory nuclei. It basically allowed us to understand that there is a possibility of de-differentiation based on the environment in which this nuclei is located, and that's how this clone was generated. That's again, without epigenetic understanding, it's not possible. So basically epigenetics help us to understand why we can't make a baby from 2 sperms or 2 eggs, but have to have one of each. It helps to understand what makes cloning possible, as we just heard, saw in the last slide. It also helps us to understand why is cloning so difficult, because as I told you, going uphill is always difficult. Why do some plants need a period of cold before they flower? So epigenetics is not just limited to humans or animals, it's also there in plants, right? Without a period of winter, some plants, they do not flower, right? And that happens because of the epigenetic changes. Why queen bee and worker bees are completely different in form and function? We just talked about this. Why are all tortoiseshell cats are female? You might have seen the cats which are having variegated skin, or the coat is having different colors, right? If you see any cat with these variegated coat, you can be certain that this cat is a female. Why? Because the coat color, it lies on the X chromosome, right? And this, as I told you, that one of the X chromosome gets inactivated at the time of embryonic development, right? So if there are 2 alleles of this coat color, say one is brown and one is yellow, or one is some other color, right? So depending on which X chromosome got inactivated at this early embryonic life, it's going to have 2 different colors when this cat becomes adult. So this is again because of the epigenetic changes you see these tortoiseshell cats. So why is that human contain trillions of cells? In the hundreds of complex organs and microscopic worms contain about 1,000 cells and only rudimentary organs, but we know that we and worm have about the same number of genes. If you look at the number of genes, we are not very different from those lower organisms, but still there are a lot of, we know that we are functionally and structurally more complex as compared to worms. And the reason for that is again the gene regulation, not the number of genes. So basically epigenetics is everywhere. It controls genes, whether you exercise in the different environment, emotional health, nutritional factors, all of these factors affect the epigenetic changes. Also, it makes us unique because at the level of genetics, we are actually quite similar, but it's the epigenetics which makes us different. And the good thing about it is reversible. That's why there is so much interest on understanding the epigenetics in humans as well as in different animals, so that we can understand how does it affect different diseases and what role does it play in different diseases, so that we can develop better treatments, better cure for various diseases. Now, how much time do I have? Okay, so I told you about the epigenetics, and in last 2 minutes that I have, I'll just tell you what we do in the lab at IISER Bhopal. So do you know that in humans, one gene can make multiple mRNAs? And that happens because of alternative splicing. What happens is there are say multiple exons. Because of the alternative splicing, different combinations of exons can be included or excluded, and they can make alternatively spliced mRNA. And these mRNAs can be translated to give rise to different protein isoforms, right? So from the same gene, you can have multiple mRNAs, multiple protein isoform. So although the number of genes are same, if you look at the humans, 20 to 25,000 genes, even the Drosophila, C. elegans, and all these different species, if you look at in the evolution, the number of genes have not changed much. The protein coding genes I'm talking about, right? But if you look at the alternative splicing events, it has gone up gradually in the evolution, and we know now that more than 90% of the human genes undergo alternative splicing. And these splicing events, they're regulated, very tightly regulated in different cells, different tissues, at different developmental stages. And if there is any deregulation that results in diseases, including cancer, where more than 15,000 cancer specific splice variants have been identified in different types of cancers. So that's what we do in our lab. We study how these alternative splicing events are deregulated in cancer cells, and how do they give rise to, how do they contribute to the different hallmarks of the cancer, be it the epithelial to mesenchymal transition or angiogenesis, autophagy, different hallmarks we studied in our lab, and try to understand how this splicing is contributing to the cancer. So because the time is limited, I think I'll stop here, and I'll be happy to take any questions that you may have.
मैं ये कहना चाहूंगा कि इतने जबरदस्त इंटरेक्टिव क्लास होने के बाद क्या हम डाइवर्स तरीके से तालियां बजाएं या एक साथ सिंक में बजाएं, या दो बार करना है और एक नया ताली कॉस्मिक क्लैप, मैं 1 2 3 बोलूंगा, आपको 3 बार बजाना है एक साथ। मैं जब 1 2 3 बोलूंगा, brain and body coordination is important, sir, which is really important for all of us. 21st century of science, but the 20th century है। 1 1 2 3 के बाद आपको करना है। 1 1 2 3 3 बार 1 2 3 1 2 3 साथ में करना चाहिए। One more time 1 2 3 थैंक यू। और आपको नींद से शायद हम तो स्लीप मोड में भी चले गए होंगे, और हमारा ये सर्वर जो है अगेन इट हैज टू फंक्शन लाइक एनीथिंग, तो सैनिटी लिमिट के साथ आपके मन में कोई प्रश्न है, सवाल है आपके? आप प्लीज थोड़ी देर के लिए हमसे भी बातें कर लीजिए, कुछ क्वेश्चन है आपके पास सबसे पूछने के लिए। अच्छा, तो सबने मैं हमारी जो ऑनलाइन ऑडियंस है उनको भी कहना चाहूंगा, इस प्रोग्राम को जो भी देख रहे हैं अंतिम विज्ञान के भोपाल का, ये साइंस संस्था के महोत्सव के दौरान आयोजित किया जा रहा है। कल हमारा आज भी प्रदर्शनी हुई है शाम के 5:30 तक, क्योंकि ऑफिशियली आप कह सकते हैं 6 बजे तक आप सब आ सकते हैं, एंजॉय करिए। बड़े बड़े वैज्ञानिक संस्थान आए हैं। कल भी हमारे बहुत बढ़िया लोकप्रिय विज्ञान व्याख्यान है आर्टिफिशियल इंटेलिजेंस के ऊपर 11 बजे, आप आइए देखते हैं हमारे ऑडिटोरियम में सुनने, और कल इसका समापन भी होगा हमारे इसी सभागार में। इसी साथ एक बार फिर से हमारे आज के स्पीकर को जोरदार तालियों के साथ बेटा थोड़ा सा खिसक जाना सर के लिए चीज हमें, सर आप यहीं रहना पड़ेगा, आपको क्षमा चाहूंगा मैं हम रख लेंगे सर, आपका सामान बस भूलिएगा। निकालो हाँ, स्वप्निल कट करो, नहीं पूछ रहे, लाइव को कट करो।